Welcome to G Fun Facts Online. Today, we are skipping the usual pleasantries and diving straight into a pivotal moment in scientific history. On a quiet Wednesday in October 2024, the world of neuroscience fundamentally changed. We are talking about the completion of the FlyWire Connectome. For the first time in human history, we possess a complete, neuron-by-neuron, synapse-by-synapse wiring diagram of an adult brain capable of complex behaviors. We are not talking about a microscopic worm anymore. We are talking about the adult female Drosophila melanogaster, the fruit fly. This is a creature that can walk, fly, navigate, fight, court, remember, and learn, and we now have the map of its mind. Let's look at the numbers, because they are staggering. This project, led by teams at Princeton, Cambridge, and the University of Vermont, mapped exactly 139,255 neurons. These neurons are connected by 54.5 million synapses. To visualize this, imagine 150 meters of electrical wiring packed into a speck of tissue smaller than a poppy seed. This is effectively the Google Maps of the brain. Just as you can trace a route from your house to the grocery store, researchers can now trace the flow of information from a fly's eye all the way to the muscles in its wings. But to understand why this is such a massive deal, we have to look at where we started. For decades, neuroscience faced a problem that project leader David Bach jokingly called neuronihilism. The brain was just seen as too complex to map. Before FlyWire, the only complete connectome we had was for a tiny roundworm called C. elegans, completed in the 1980s. That worm has exactly 302 neurons. It doesn't have eyes, it doesn't fly, and it doesn't make complex decisions. The fruit fly brain is 500 times larger. Mapping it is equivalent to mapping every road, alleyway, and driveway on Earth, down to the resolution of a few inches. So how did they do it? It started in 2018 at the Genelia Research Campus. They took the brain of a single female fruit fly and sliced it into 7,000 ultra-thin sections using a diamond knife. Each slice was only 40 nanometers thick, which is about 1,000th of the width of a human hair. They imaged these slices using high-speed electron microscopy, generating 21 million images. The total dataset was 106 terabytes. If you tried to download that on your home internet, it would take years. But having the images was just step one. In these grayscale images, neurons look like spaghetti tangled in a bowl. You have to trace one strand of spaghetti through thousands of slices without accidentally jumping to a neighboring strand. If a single human tried to do this manually for all 139,000 neurons, it would have taken roughly 4,000 years. The solution was a hybrid of artificial intelligence and human effort. The heavy lifting was done by AI models developed by Sebastian Soon's lab at Princeton University. They used convolutional neural networks to scan the images and generate 3D reconstructions, effectively coloring in the spaghetti strands. But AI isn't perfect. It makes mistakes. It might merge two neurons that touch or break one neuron into two. In a circuit diagram, a single error is catastrophic, like connecting a power line to a water pipe. This is where the project truly revolutionized science. They gamified the process. They built FlyWire, an open, browser-based platform that allowed anyone in the world to log in and fix the AI's errors. This used a chunked graph architecture, allowing for real-time collaboration. If someone in Tokyo fixed a neuron, someone in New York saw it instantly. This brings us to the human heart of this project. Among the citizen scientists, one name became legendary: Christoph Kruk. Kruk isn't a professional neuroscientist. He is a 40-year-old electrical engineering graduate from Poland who battles generalized anxiety disorder and social phobia. For him, the repetitive, detailed work of tracing neurons was a meditative escape. He told reporters that it allowed him to do science without the administrative burden or the feeling of wasting time. While most participants traced a few dozen cells, Crook personally proofread and annotated nearly 50,000 neurons. That is more than a third of the entire brain. His contribution was so massive that he was listed as a co-author on the flagship Nature paper. Collectively, this army of researchers and gamers made over 3 million edits to the dataset, turning a raw map into a labeled atlas. So what did they find? The FlyWire project revealed that the fly brain contains 8,453 distinct cell types. Of those, 4,581 were completely new to science. It's like trying to understand a car engine when you only know what a tire looks like, and suddenly finding the spark plugs and transmission gears. Network analysis also revealed that the brain is an oligarchy, not a democracy. There is a rich club of neurons, a small percentage of cells that are highly connected to each other, acting as high-speed superhighways for information. They also identified two fascinating classes of hub neurons: interrogators and broadcasters. Interrogator neurons are like spies. They have inputs reaching into diverse parts of the brain to gather disparate information, visual data, smell, hunger levels, to form a global picture. Broadcaster neurons are the commanders. They send signals across massive sections of the brain to coordinate global state changes, like signaling danger or mating mode. Perhaps most importantly, they debunked the snowflake myth. There was a fear that every fly brain might be unique, but by comparing the FlyWire map to a partial map from a different fly, they found high consistency or stereotypy. If neuron A connects to neuron B in one fly, it almost certainly does so in another. This confirms the brain is a hardwired machine built on a genetic blueprint. But a map is only useful if you can navigate it. The FlyWire papers included several proof-of-concept discoveries that explain actual behavior. Let's talk about flight. Flies are master aviators, and we now know why. It involves the ocelli, the three simple eyes on top of the fly's head. These aren't for seeing images. They are motion sensors that detect the horizon. Researchers mapped a circuit involving neurons like OCG01 that take input from these eyes and wire directly into the descending neurons connected to flight muscles. It is a reflex arc that bypasses the thinking brain. If the fly tilts, the ocelli fire, and the wings adjust in milliseconds. It is an onboard autopilot. Then there is the walking circuit. How does a fly stop walking? It turns out there are two ways. There is a walk-off mechanism, which uses GABAergic inhibitory neurons to cut the power to the walking commands. It's like taking your foot off the gas, used when the fly stops to eat. But there is also a brake mechanism. This uses excitatory cholinergic neurons in the nerve cord to actively stiffen the leg joints, locking them in place. This is like slamming on the emergency brake, used when the fly needs to groom itself. In the visual system, which takes up 60% of the fly's neurons, they mapped the lobula plate tangential cells. These calculate optic flow. They found neurons that specifically fire only when the visual field rotates clockwise, or only when the visual field expands, indicating an impending crash. They even mapped the circuitry for color vision, showing how interneurons compare signals from UV-sensitive and green-sensitive photoreceptors to create a color opponent channel, basically biological RGB sensors. Now you might ask, why does this matter for humans? We share 60% of our DNA with Drosophila. More critically, 75% of the genes that cause human disease have a counterpart in the fruit fly. Many neurological disorders like Alzheimer's and Parkinson's are increasingly viewed as connectopathies or diseases of wiring. With FlyWire, we have a baseline for a healthy brain. We can now create genetically modified flies with Alzheimer's traits and map their brains to see exactly which circuits fail. Does the disease attack the rich club hubs first? Do specific synapses degrade? For Parkinson's, which affects dopamine, FlyWire has mapped every dopamine-producing neuron. We can trace exactly what they connect to, helping us understand the motor loops that degenerate in humans. We are moving from a connectome to what scientist Mala Murthy calls an effectome. Because we know which synapses are excitatory and which are inhibitory, we can simulate the brain. Researchers are already building in silico fly brains. They can input a digital visual stimulus, like a predator, and watch how the virtual neurons fire. If the simulation predicts a jump and the real fly jumps, we have effectively uploaded a piece of a biological mind into a computer. This is the future testing ground for drugs. The FlyWire data is free and open to the public via the FlyWire Codex. It operates like a search engine for the brain. You can ask it to show you all dopamine neurons in the right hemisphere, or trace the path of light from a photoreceptor. High school students can now do research that was impossible a decade ago. Looking forward, this is our Wright brothers moment. The Wright Flyer didn't go to the moon, but it proved flight was possible. The scientific community is now pivoting to the mouse brain, which has 100 million neurons, 1,000 times larger than the fly. Projects are already underway to map a cubic millimeter of mouse cortex. Mapping a human brain with 86 billion neurons is still science fiction, requiring zettabytes of data. But we don't need a full human map to learn. By understanding the fundamental principles in the fly, how interrogators work, how brakes function, how the rich club coordinates states, we can infer the architecture of the human mind. The FlyWire Connectome is a cathedral of data, built by thousands of invisible hands, from tenured professors to a gamer in Poland. We have finally lifted the hood of the machine. As we look at the wiring of the fruit fly, we are seeing the physical shape of a decision, the geometry of a memory, and the architecture of behavior. The fly has landed, and the age of the connectome has officially begun. Thank you for listening to G Fun Facts Online, and make sure to check out the codex yourself to see the wires that make life move.